Welcome to the 2014 cancer class. This is just going to be kind of your first day in introduction to the disease of cancer. Now, it's one that I have a lot of interest in and a lot of respect for. What I'm going to show you just brushes the surface. There's a lot more to this disease than what I'm going to show you today. Now, we're going to spend the rest of the semester figuring out exactly how it does what it does, but I thought that I would just give you a basic introduction so that we could all start on the same page. The most interesting thing that I think, the thing I find the most interesting about cancer is that it's not a disease that you catch. It's not something that you get from like an STD or from a mosquito. The whole of the disease is contained within the information that we have, right? So our cells all have information about how to grow, when to divide, how to make things. And cancer, the information for cancer is actually in that same kind of genetic coding. And I think that might be the biggest problem with cancer, is that unlike other diseases, what we're trying to fight is basically our own cell. The information that makes a cancer cell different than a regular cell doesn't always change the appearance of the cell. So when you look at two cells, one a cancer cell and one a regular cell, you don't always see any difference. And that's the biggest problem with attacking it as well. If you're hunting cancer cells, it's very hard not to hunt regular human cells as well. I particularly like this quote from Sun Tzu, and that's, know thy enemy and know thyself and you will never lose a battle. I think it's kind of the key to studying cancer. By studying cancer, you have to learn about our own processes, and that's what we're about to do. So, at the heart of the whole disease is basically a system of checks that our cells go through. Now, you start as one cell and then you become a trillion, your entire body. Your cells must divide many times, but they can't divide uncontrollably. So, for example, a cell in your forehead, if it started dividing uncontrollably, you would end up with like a tumor on your forehead. If something like that happened in your eye or brain, you would end up with a tumor there, and the results would be really, really bad. So, cells actually have checks. They have reasons to grow, and they have a system which measures whether or not the conditions around it are optimal, and then make the decision to grow or not to grow. Now that's all again in the DNA, in the genetic coding. So we're going to go through a few of those checks, and we're going to see how making those checks a little bit different can actually cause a problem. In addition to making sure that you just don't have uncontrolled growth and tumor growth, the other reason, or the other advantage to having these checkpoints is that cells that are damaged or with changed information, with maybe mutant information, are destroyed. Because then they can take, if they aren't, then they can take over the cell. So, I'm not sure this is always considered a checkpoint, but I think it's one of the most important aspects of the cell cycle, or the process by which a cell commits to dividing and then begins to actually divide, splits its DNA, or copies its DNA, splits it, and then divides. The first thing, and the simplest thing, is that you need a growth signal. Now, humans are unlike bacteria. They're multicellular organisms, and those cells must coordinate and communicate with each other in order to survive. Bacteria do something similar, don't get me wrong, I'm not saying that they're uninteresting, but human cells specifically need a signal to begin the growth cycle. They need a signal for them to start committing the resources to growing, copying that DNA, splitting that DNA, and then splitting the cells apart. They also have a process by which cells can check the DNA. If you have DNA, you know that T's line up with A's, G's line up with C's, and so on and so forth. And when those line up, that generally means that you've copied DNA successfully. However, sometimes if you miscopy DNA, so for example you make a T with an A and then a G with a G, what you'll sometimes find is that the DNA doesn't line up as nicely. And when the cell can detect that, it can either try to to destroy that cell, or it can attempt to fix that DNA. If it can't fix the DNA, the cell will commit to something called apoptosis, or suicide. The idea there is kind of like it's best to just destroy the thing that might hurt us completely than being a little more cautious and allowing it to get out. There's also something interesting about most cells in the human body. A lot of people don't know this, but there's something called reproductive quota. And most cells in the human body can't just divide in a million times. They have a fuse or a timer that says you can only divide this amount of times before it stops working. And we're going to go through that. So you may or may not have seen this process before in your classes. This is the process of replication. And it is one of the main ways that both cancer arrives and cells divide. Again, it's kind of that dichotomy about the disease really being necessary for life. So in this process, DNA is fed through a machine and it's copied. Now you have 6 feet of DNA, a billion-ish base pairs, and each of those needs to be copied pretty accurately. The whole process is remarkably accurate. When mistakes occur in that copying, mutations occur, and depending on where those mistakes are, those mutations can generally be disastrous. Most of the time, they're not going to be that big of a deal. But when precisely placed, when the mistake is just at a very, very, very important part of the genetic information, then you can end up with some big problems. Now, I said that cancer cells in the body need to start with a growth signal. That's often called a mitogen. Some people mistake that for a growth factor or a growth hormone. There's a little bit of a difference. It's not that big of a deal. But essentially, in order for this cell right here to begin the division process, it's going to need a signal designated by this thing in purple that's going to bind to a receptor designated by this thing in green. That purple thing is going to line up to that green thing at those specific contact points, and it's just a whole positive versus negative thing. They're going to line up, and then that cell is going to have something happen. The receptor, this purple thing, is going to communicate the fact that it bound to something to the inside of the cell. So let me make this a little clearer. Let's say we had a signal bind here, or here. Well, the receptor has a part inside of the cell that is going to activate a protein, which is going to activate another protein, which is going to activate another protein, which is going to go and interact with DNA. Now here we see that interaction with DNA. This is DNA here. On top of it, you have something here called a transcription factor, or sometimes an initiation factor, depending on the organism and the complexity you're describing it in. That thing allows RNA polymerase, a copying device, to bind to that DNA right there. And when that RNA polymerase is activated, as you're about to see, I always get the timing wrong, so we're going to try to get this right, but I can't guarantee it will be. But the RNA polymerase will begin to copy a piece of the DNA. And depending on where that transcription factor binds, and then starts reading in 3, 2, 1, screwed up. Uncomfortable silence. And starts copying, that's how we determine what proteins are going to be made. So again, running through the whole process, something binds to a receptor on the outside of the cell. The receptor causes a change on the inside of the cell. That change activates a protein, which activates a protein, which activates a protein, which then goes to DNA, allows RNA polymerase to copy a small segment of that molecule. This is the copy here in a process called transcription. And then results in a mRNA transcript, which will go to a ribosome and then be copied. Now, cancer cells somehow figured out a way around this. Now, and figured out isn't exactly the right way to think of this. It's more like their coding instructions changed. So a cancer cell's coding instruction changes in a way such that many of them no longer require that signal. Or they become hypersensitive. So let's say this is a normal growth signal. You have one molecule binding to a cell. Cancer cells figure out many ways around this. They can become incredibly sensitive to that one signal. Or they could in fact get cells around them to produce a lot of growth signals, causing this cell to divide. There are also don't grow signals. So cells, when they come in contact with these don't grow signals, oftentimes shut down. The programming of a cancer cell, many times though, allows it to completely ignore these don't grow signals. So let's just look at what we have. Cancer is caused by changes in the genetic code. Those changes in genetic code happen just because we replicate DNA. That change in DNA can change the programming of a cancer cell such that it no longer requires a growth signal or becomes very sensitive to a growth signal, or it becomes insensitive to a don't grow signal. In that way, they kind of bypass that first checkpoint. They don't require growth signals. Now, DNA checking, that's another really important thing. Remember I said when you have a mutation, sometimes the DNA bows out. It forms like almost like a bubble. So you'll have two strands lined up perfectly, and then two bases won't line up, and one piece of the DNA will bow out just a little bit. Proteins can go around and check that for that bowing and then try to repair it. This protein is called P53. Now I'm going to oversimplify this, so don't shoot me here. But basically the way it works is that DNA has a phosphate group on the outside of it. It's negatively charged. This protein has a positively charged part here. That's why the protein attracts the DNA. The DNA is negative, that part of the protein is positive. Now that P53, that protein, is one of the things in the machinery that allows DNA to correct itself. Now what if this piece right here, this P53, that piece, that one positive charge that allows it to bind to that negative charge on DNA, were charged something different? What if it was neutral? Or what if it was negatively charged? Then you wouldn't have that same binding. At the end of the day, biology is just pluses and minuses coming together. And if you don't have a plus and a minus, two things don't come together. And if this thing is responsible or plays a part in DNA checking, then all of a sudden, you've really, really, really limited your ability to check your own DNA. Now, if you've limited your ability to check your own DNA, you've also limited your ability to repair it, and you've also limited your ability to commit suicide in case there's too much damage. That's a crazy idea. Suicide, death, is something that cells, our cells, actually acquire the ability to do. It's a process they gain. So we are always kind of like involved in this search for immortality when in actuality evolution has given us death. Just kind of an interesting aside. But when you destroy or damage your ability to check your own DNA, you've also, in a sense, hindered your ability to commit suicide. Now remember I said that cells only divide a certain number of times, or most cells only divide a certain number of times? Well, that's because every time you copy DNA, it becomes a little bit shorter. So if I were to take DNA and I were to clump it up, twist it up really, really tightly, it would form these almost rod-like structures called chromosomes, and that's what we're looking at here. So those are pieces of DNA, and every time DNA is copied to produce another cell, it gets a little shorter. So this is the DNA from the original cell, this would be a copy made, and this would be another copy made. What if there was an extremely, extremely important protein right here? Well, this copy has it, this copy has it, but guess what? This one doesn't. So, oversimplified again, that's kind of the way that cells have timers. When they run out of, or run into that problem of not copying that important DNA, cells stop being able to replicate. Now, cancer has evolved, again, by just changing its programming just a little bit, it's evolved a way to bypass that problem. And what it sometimes does is sticks these little pieces of DNA, and I don't know why, but I've signified them as Chuck Norris. But by sticking little pieces of nonsense DNA on the ends of these chromosomes. That means that instead of destroying this piece here, we would have destroyed a bogus piece. basically destroying the cell's ability to have that timing effect. It's an interesting disease, it's a creative disease, and what we've just seen are some of the ways that the programming can bypass the requirement for growth signals, or make them super sensitive to growth signals, or make them insensitive to not growth signals. We've seen how you can pretty simply limit the ability to check DNA, and because you've limited the ability to check DNA, you've limited the ability to repair DNA, and therefore the ability to commit suicide in case you find damaged DNA. And now we find that you've knocked out some of the ability of this cell to time the number of times it divides. It's an amazing disease. It's no wonder that almost every academic institution in the United States is working on it. But that's not it. We're going to study cancer for an entire semester. That's all we're going to do. But the problem is much bigger than that. Cancer cells are incredible in what they've evolved to be able to do. Many of them, in fact, increase blood flow to themselves. So they produce something called angiogenic signals. Let's say you have tumor cells here. Well, they're using a lot of energy. And energy is, as we know, carried in the form of glucose in the body. And glucose is carried in blood. So these cells have actually acquired the ability to increase the blood flow to them. They will get the body to build its own blood system. On top of that, there's two ways of utilizing energy in the body. There's a really more efficient way called aerobic respiration, and then there's an inefficient way called anaerobic respiration, which produces lactic acid. You know it, it's what causes the burns in your legs when you're lifting, when you're doing something that requires a lot of energy really quickly. You would think that a cancer cell would acquire the ability to use a lot of energy extremely efficiently. But it turns out that's not always the case. Sometimes cancer cells revert to an inefficient form of energy use, which means that they burn a ton of energy but don't utilize all of it. What that means is that this rapidly dividing cancer cell, this cancer cell that's doing all the things that we just talked about, is using a lot more energy than it needs. And what can happen, is that the area around the cell becomes like a desert as far as energy is concerned. And it oftentimes becomes a desert as far as other nutrients like oxygen are concerned. The cancer cells starve the cells around them. It's important to stop thinking about cancer in a couple ways. First of all, cancer cell is not a single cell, never. Cancer cell is never ever just a single cell. A cancer cell is a group of cells, an environment, many different types of cells that are interacting with each other and helping each other out to form an environment. Cancer is really a microenvironment that allows tumors to form. I think it's also important to remember that cancer doesn't try to evade checkpoints. It doesn't creatively do anything. Though I use those words, what I'm really just doing is paying homage to what happens. This is all programmed. It's important to realize that all of this that I've described occurs in the coding level first. So all the abilities I've talked about, that change in abilities starts with a change in the structure of the DNA. All the time, every time. It's always structure change, then function change. And that's important to remember. This is what we're going to be studying for the entire semester. I think this is incredibly fascinating, and it's certainly worthwhile. There are a lot of people suffering from this disease. I bet you, if you asked any person you know, almost every single one of them have had a person either affected by cancer or are affected by cancer themselves. Good luck this semester. It's going to be tough, but it's going to be rewarding.